Okay, so this video is going to be about codominance, which kind of does what it says on the tin, really. So alleles can interact in different ways. So we've done quite a few crosses where we've talked about dominant alleles being expressed in both the homozygous and the heterozygous state, and recessive alleles only being expressed in the homozygous state. With codominance, what that actually means is that both alleles are expressed. So that means they show up phenotypically if they're present. Which leads to some sort of, these quite interesting scenarios. So I'm going to deal with the simplest ones first. So if you imagine you've got a plant with a red flower, and this particularly applies to snapdragons actually, but I can't draw snapdragons, so I'm not even going to try. So we've got a red flowered plant, and then we've got one with white flowers. And if we cross them together, what we find is that we get pink flowers. So that's sort of like almost an intermediate blending sort of inheritance. Now why that happens is that what's going on genetically, so these are the phenotypes, is that we have two dominant alleles causing the red flower, we have two dominant alleles causing the white flower. These ones are making only R-type gametes, these ones are making only W-type gametes. You stick them in a Punnett square, parent 1, parent 2, and these pink flowers have the genotype RW. So the heterozygotes are giving you a completely different phenotype. Now if we, so these are the parent generation, these are our F1, and if we go through to the F2 generation, what we're crossing together is two of these plants. So they both have the genotype RW. Now that means that they can make two sorts of gametes. They can make ones with the R allele in, ones with the W allele in. This one the same, ones with the R allele, ones with the W allele. And when we put them into the Punnett square, parent 1, parent 2, we get RR, RW, RW, WW. Now this is the only time that we ever really use two different letters and in capitals for one characteristic, which in this case is flower colour. So our RR1 will be red, and our RW ones, both of them, the heterozygotes, will be pink. So you can see that this is kind of a bit of a sort of variation on that 3-to-1 ratio. So we've still only got 4 offspring. Normally we'd expect in dominance recessive crosses to get 3 dominants to 1 recessive. Now we're getting a 1-to-2-to-1 ratio, and here are phenotypes: red, 2 pinks to white. And there are a variety of sort of animal crosses that do more or less the same thing. The pink colour in animals is called roan, so you'll see that word quite a bit. So yeah, slightly different ratio. Just to take you to a human example then, there is a condition called sickle cell anemia, where the red blood cells of somebody that doesn't have any of the sickle cell alleles is sort of donut shape, and if you have the sickle allele, the red blood cells, actually the protein changes shape at low oxygen concentrations and gives that sort of sickle moon kind of shape. So, and these two alleles, so this is a mutation, are codominant. Now, oddly, in blood cells, we refer to the allele as HB rather than just giving it a single letter, HB standing for hemoglobin. So somebody with normal blood would have two normal alleles, HB, HB. Sickle cell, you could have HB, HBS. This is the heterozygote. Or you could have HBS, HBS, which is the homozygote. And we say that these have got sickle cell trait. Now that means it isn't quite 50-50, but you can think of it as 50-50. It's more like 60-40. It means that 50% of the cells are normal. So that means that that allele is fully expressed into 50% of the cells, and 50% of the cells are sickle. So half of the cells are expressing that allele. And of course in HBS, HBS, no choice, you've got then sickle cell anemia, which means all cells can sickle. And you think, well, that's really quite deleterious, because if they're sickle shaped, they don't fit, they can't squeeze through those capillaries. This causes crippling joint pain, amongst other things, and obviously a failure to carry hemoglobin. But actually, this genotype here is quite advantageous in malarial countries. So these 50% sickle cells are not able to be invaded by the malarial parasite, so they offer a bit of protection from malaria. Now obviously, if somebody with normal hemoglobin goes to a malarial country and catches malaria, it can be fatal. It's still one of the biggest killers worldwide, and so that's a disadvantage. This is going to be a disadvantage wherever you are, because of the crippling joint pain and anemia, having both alleles. But this one is an advantage, so you'll also see this when we do evolution, probably, called a heterozygote or a heterozygous advantage. Now this allele is just inherited in the normal way, so where we've got high incidences of this sickle cell trait, you'll get a lot of the matings will be between two people who have sickle cell trait. They will produce each two types of gametes, one with the normal allele in and one with this mutated allele in. And I think you can probably predict already what's going to happen when I stick those into my Punnett square. So every time two heterozygotes have a child, there's one that has normal hemoglobin, there's one that has sickle cell trait, this one has sickle cell trait, and this one has sickle cell anemia. So this one really disadvantageous, this one disadvantageous in malarial countries, and these two are going to be okay if there's malaria present. So we've got, each time a couple that are both sickle cell trait have a child, they have a 25% chance of their child having normal haemoglobin, a 50% chance, because it's a 1 to 2 to 1 ratio, of them having sickle trait, and a 25% chance of having a child with sickle cell anemia. So a quarter. But that is the same every time, so there would be, just like there are families full of girls or families full of boys, there would be nothing to say that an entire family might have child after child after child with full-blown sickle cell anemia, or child after child after child with normal haemoglobin. That's just the nature of chance. It's a probability. It's a 25% chance each time you have a child. It doesn't matter how many children you've already got that have, say, sickle cell trait. There's always a chance that the next one, there's a 1 in 4 chance that the next one will have sickle cell anemia or will have normal haemoglobin. That's just the way it works. I'm going to do blood groups, I think, next as an example of codominance and recessiveness sort of combined, because that's a bit more complicated.